Hello.
Hello, welcome to stream number, I'm going to go with 3.
I think that's true.
Yeah, number 3 of our Science Scribbler Q&A and Classify.
I thought you were gonna give a denominator.
Number 3.0, 3.0, 3 out of O.
Exactly.
3 out of infinity.
Yeah. Yeah, it's great to have you here. We're excited. I'm Michelle.
I'm Trisha. Yeah.
We are part of the Science Scribbler team. And today we are going to choose a random project and do some classifications on stream while we chat.
Yep. Okay, so I'm going to share your screen so that we can pick a project together.
Yes, please do.
Alrighty. There we go. So what are we feeling today?
You had two that you were talking about earlier.
Yes. So I think maybe we should start with Notes of Nature California one. I think it's pretty high up on the homepage of projects.
Okay.
There we go. Notes from Nature. Cas plants to pixels. I think I got attracted to this because it said pixels.
Oh, okay.
Yeah.
And you were like, I know pixels.
Yeah. and that the homepage was so pretty.
Oh yeah, that's a beautiful photo. These images hold tons of critical information about California plants, where they grow, what they look like, when they flower and fruit, and we need your help unlocking it. Okay. So it looks like they are 68% complete on one of their workflows, 0% on another one, and then 30% on the third.
Yep.
Where are we feeling?
I'm not sure where we're feeling, but I guess one thing also to note from this is that it's part of a wider Notes from Nature organization. So this project forms one of loads that are going on all the time, all centered around, I think, the same kind of archive images. Yeah.
Oh, cool. Okay, yeah. I didn't realize that. I was expecting beautiful photos of like researchers' hands cupping, and we got to say what plant it was. Okay, so we're not doing that.
I think we're looking at maybe pressed flowers.
Yeah,
or like
now
pressed
that
plants.
now that I've scrolled past these pictures here, I think you are correct.
Yeah.
Oh, that's funny.
But
okay.
Still pretty.
Yes, still pretty. Just maybe slightly less vibrant. Okay. And let's just read this about Notes from Nature. Our collection of California plants dates back to 1853, an extensive record of where plants have lived across California through time. Understanding where plants live now and in the past is key to understanding their biology, their evolution, and how best to protect them in the face of climate change. Okay, so they're digitizing the California specimens from a museum, I assume, of some kind.
Yeah, and I think that the Notes from Nature team generally are about digitizing these kinds of museum records.
Okay. Okay, cool. It says we've scanned high quality images of our specimens and we need help transcribing the information on those images into digital text. Okay.
Ooh, so it might be a transcription task.
I was going to say, I think this might be transcription rather than...
Let's have a look and see whether it's...
That's fine.
Yeah.
The only, not to say that we don't like transcription tasks, because we do. It's just that we have a touchscreen tablet and a pen, and those are not the right tools for transcription tests. So we might need to bail, and instead, when we choose to do it, we can set it up with the computer rather than the touchscreen. But let's have a look at it and just see. So let's do the 68% complete one, because I think it's always fun to be part of like the tail end of a project as it's like speeding up and almost done. So. Okay.
Oh no, it is transcription.
Okay.
We're gonna have to push this to the next one where we will bring a keyboard rather than a drawing tablet.
Yes. Yeah. Okay, cool. I just want to see. Oh, they don't even have any images in. Okay. Field guide. Ooh, the Getty Thesaurus of Geographic Names. That sounds fun. That sounds like a fun tool to learn more about. But I think we might be doing this next time. Yeah. Okay, cool. So what was your second choice?
My second choice was Monkey Health Explorer.
Monkey Health Explorer. Yeah. Okay, cool. Let's go back to projects. And do you think we'll have to search for that one?
If we go into biology, it's probably going to come up quite early.
I like this frog find picture.
Yeah, so I really like that too. And when I was looking for a project today, I clicked through, but it's actually an audio-based project,
Oh,
listening to the rivets,
the croaks.
yeah, of
Yeah,
frogs.
that
And
sounds really fun. Okay, well maybe next time. Well,
yeah,
2 times from now.
I think we need to check whether or not we can successfully listen to audio while live streaming. I'm not sure that that's possible.
That's fair. We can do some testing.
Yeah.
Look, it's ours. Science Scribbler Placenta Profile. Slight plug. Sorry. Where's this monkey one? Monkey Health Explorer. Oh,
Yeah.
it's a cute little one. I'm expecting a cartoon monkey. Help us learn more about monkey health by identifying white blood cell types.
Yeah.
Okay, and they say single cell 2 is our top priority. Thanks for your awesome help. Okay, so maybe we will focus on single cell 2.
Have you contributed to this project before?
I have not.
I have, but before the single cell workflows were there, and so I think it's a blood smear.
Yeah.
That you're looking for white blood cells in.
Okay.
But it looks like they've progressed their project, so they're now asking people to identify a single immune cell that has already been found within the smear, rather than looking at the whole smear and getting people to identify.
Got it.
Each cell.
Okay.
Yeah.
All right. Well, so they have a clear starting point. They say beginners practice. Start here. So that's how we identify white blood cells, and then after that we can start going to the single cell 2 or some of the other ones. But let's start where they want us to start, with the practice. Okay, so they've got 5 overlapping images here showing us different cell types. So in this project we ask you to look at images of cells and identify the white blood cells by type using example images in the field guide. So neutrophil, lymphocyte, monocyte, eosinophil, and basophil. And there's going to be a bunch of red blood cells as well, and platelets, and some cellular debris that they don't care about. So let's look at this a little bit. So I actually did a project in grad school on red blood cells, so I know intimately their shapes.
Did you also have to look at white blood cells during your project of red blood cells?
Those were like a contaminant for me.
Okay.
Because I was interested in sickle cell, and so that's something that affects the red blood cells exclusively. And so when I found other cell types, it was annoying. But okay, so we've got very, very tiny platelet. We've got neutrophil, monocyte, basophil, lymphocyte, and eosinophil. Okay. Oh, this is really nice. I love it when tasks explain how to actually use the tools that you have access to, because sometimes it can be a bit unintuitive. So it looks like we have our marker, our pan, zoom in, zoom out, rotate, and reset back to where we were before. And then they're also pointing out the field guide and the help text. Okay, so Goldilocks test. Before marking white blood cells in some workflows, you will be asked to look at the overall spread of the cells to determine if they are too sparse or too thick to count. If in doubt, mark the spread as good. There is also an option to mark an image as low quality if it is too blurry or has too many debris. Okay, that's nice that they tell you, like, if you're unsure, to mark it as good. It's useful, kind of helpful information. How to mark white blood cells. For each white blood cell in the image, click on the cell to mark it. Choose the cell type from the list. Use the field guide to see examples. Some cells won't exactly fit the description or match the guides, so make your best guess. Do mark cells cut off at the image edge if you can identify the cell type. Okay, again, really helpful instructions. They've done a good job here. Okay, and then there's a video tutorial, which might help us to understand for a future monkey-based project.
Yeah.
So I'm going to hit play and we're just going to let it sit for 3 seconds.
I don't know whether...
You don't know whether...
I don't know whether we can play someone else's YouTube video on a live stream.
Well, if it's YouTube, it's...
Is it public?
Yeah.
Okay.
And also, like, we're adding... What are the what are the laws? I think because it'll be so short, it's fine. So.
Okay, but what's not going to happen is any audio is going to play.
Well, then that defeats the purpose.
Yeah, well, I haven't set this up right then.
No, don't bother to redo it. That's fine.
It might actually create music, but no, Zoom is muted.
It's fine.
Yeah.
It's fine. Okay. When you're done identifying all white blood cells on an image, zoom all the way out to be sure you haven't missed anything. Click Next so that you can check your work, and then click on the circle at the bottom of the image to see the answer key and tell us how you did. Click Done or Done and Talk if you have feedback.
So this is one thing that is not in very many projects, but actually people do sometimes ask for a tutorial, which is rather than the tutorial we've just looked at, it's like a small workflow where they give you the answer to kind of build confidence, I guess, so that you know that you're learning and you're identifying the right things.
Yep. Okay, so white blood cell examples, because these are the things that we're identifying. So, okay, so those are our options again. So there are 5 of them. And I always find it useful to see like the cartoon, which they've put in the middle for each of these. And then they've got some examples on either side, which is nice. So neutrophils, most common, 2 to 3 times larger than a red blood cell. They've done such a good job. I love the fact that they've given so much information, especially relative to something that they know will be very present in the image. This is really well set up. Okay, so then light pinkish with small granules and an occasional vacuole, and then the nucleus is dark purple with multiple connected shapes. The lymphocytes, light, typically bluish cytoplasm. It looks like much smaller amount. The nucleus is slightly larger than the red blood cell, and it is dark purple and rounded. So then the monocytes, these are less common, 2 to 3 times larger than a red blood cell, and it looks like the main difference is that they're kind of C-shaped or bean-shaped nucleus rather than the connected one that we had up there. The eosinophil, darker red or purplish in the cytoplasm with some granules. That's those dots, I think. And then we've got 2 or 3 round connected shapes as our nucleus. And then the basophil, very rare. Ooh, so maybe we'll have to see if we get one. Dark purple, bean-shaped, mostly hidden by granules, so darker. Okay, cool. I think that's sufficient information for us to kind of get started. So just looking at this image over on the left, I think, we've got 2 clear things up here. 1 here, 2 here, 1 there, and 1 there. And then we might need to have a bit of a chat about this situation down in the left.
Whatever that is.
Yeah. But I think that might be it. There's something going on here too, but I don't know if that fits into any of the profiles that we've just seen. So should we have a go at trying to identify which one's which now, just before we click? Because I am conscious that I'll have to close the field guide in order to click and engage.
I think we can. Yeah. What are you going for? I think what's really useful as well is that it's showing us how likely we are to see those cells as well.
Yeah.
Because that means that perhaps we can think, is it more likely to be a neutrophil?
Yeah. Yeah, absolutely. So I think the neutrophil makes sense for this one here.
Yep.
And I think for this one here. And probably these 2 as well. And then I think this one's going to be a lymphocyte.
Yeah.
This one I think might be a monocyte. How are we feeling about that? Doesn't quite have the right coloring, maybe.
Yeah, I think that some of them feel a bit trickier. So the ones that we've already identified clearly as, or we think are neutrophils, have quite a clear cytoplasm, like light pink, whereas some of these other ones are getting towards like purple or with granules. So I think that does push us further down.
Yeah. Okay, well, let's do the ones that I think were clear. So let's start with the neutrophils, because there were 4 of those, I think, that were clear. So mark them. We can zoom in a little bit. Okay, oh good, they put the little cartoons here as well. That's really helpful. Oh, but you have to scroll down in order to click OK. A little bit sad.
You might be able to zoom out on the tablet.
It's like just barely good enough, so let's see. Step is required. I don't even know how to scroll down now because thank you. Okay. Good. And then I think we also said these two on the left here. See, but their backgrounds are not nearly as like translucent pink. So like, let's have a look again at the field guide.
I think the one that I definitely can't pronounce, the eosinophil.
Eosinophil.
Yeah. That's I would be tempted by that one for those two.
Okay.
Because I think that there are probably granules visible there.
Yeah, no, I think that's a good shout because you can kind of see it. Like it looks very similar to that one. Okay, so let's mark those two as eosinophils. Do you want to practice saying it?
Eosinophil.
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 Yeah. You got it.
Yeah.
Good. Okay, I feel all right about those. We knew this one from the get-go. That one's a lymphocyte. That's good. Okay, and then let's move around a little bit. Yeah, this one over here. So I think that takes us back to, what was it, lymphocyte? Or no?
Neutrophil.
Neutrophil, that was it, at the top. Good. Oh, didn't see this one on the far bottom there. We'll have to see if we can identify that when we get to it. Yeah, there we go. Okay, so do we want to, let's zoom in and see what's happening there real quick and just kind of decide what we want to do about it. That looks kind of like two things that have maybe exploded or merged. I don't know.
Yeah, I think that that's probably going to be some form of debris.
Yeah.
On the smear. Is there a section in the field guide which talks about things that aren't white blood cells?
Not a white blood cell.
The importance of having examples that are things and also examples that aren't things.
Yeah.
Negative examples of what not to click on.
Yeah, absolutely.
Yeah.
Yeah, okay, cool. Yes, cell fakes. These are red blood cells with platelets or other debris on top, making them look like neutrophils. I think that's probably where we're at here. Okay, so let's leave those alone. Let's zoom out a little bit. Okay, so that quadrant looks good. That one looks good. What do we feel about this?
I feel that that is not a white blood cell.
Okay.
And so I think it probably is going to be some other debris.
Yeah.
And
this one too looks like debris on top of something.
Yeah. Or if it isn't, it's a burst white blood cell, which we're not interested in marking.
Yeah.
As well.
Okay, cool. So just this one. So let's look again at the field guide. Okay. Monocyte or basophil, but it's not purple enough to be a basophil. Ooh, monocyte's fairly purple too.
I would say that it's closer in terms of the nucleus. The shape of the nucleus is closer to a monocyte than to a basophil.
Yeah, I think the coloring is off too, because like the basophil, you can barely distinguish between the nucleus and the background, and like there's a clear distinguishing line here. So I don't think it's basophil.
And it's not got the distinct lobes of the nucleus of the eosinophil.
Yeah, great job. Yeah. You don't think it's like a weird lymphocyte, do you? Because there are some like kind of globular nature to at least one of these examples. And they don't take up the entire space.
I think that it's too big to be a lymphocyte.
Oh, yeah. No, you're right. Yeah, because the nucleus is about the same size as the red blood cells. Okay, so by process of elimination, I think monocyte makes sense. There we go. Okay. Let's zoom out and just make sure we haven't missed anything. Cool. And then how do we tell if we were right? Do we have to wait until the very end for that, or?
I think that if you click next, if I remember correctly, we might be given the answers.
Oh, okay. Cool.
Check your answers on the second image. Yes, so there is actually an image flipbook that we're seeing.
Okay, cool. Yeah, that was not clear. Okay, that was not the right thing to click apparently.
Okie dokie. All right.
Okay, so yeah, neutrophil, 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 eosinophil, which I think, did we get those?
I think so.
Okay. And then we definitely got the lymphocytes and monocytes. So yeah, I think we got all of those right.
Nice.
Yeah. Okay. How many are there in the tutorial?
I don't know, but I actually feel fairly confident that we can probably begin contributing to the main project.
How
only
terrible.
Because I've done this tutorial already.
Okay. All right. Well, let's skip out of this then, because that's the downside of having one of these tutorials, is that like you are taking volunteer time and effort away from your actual things to classify. So what I do think might be useful though is let's just click next and see what they mark. I expected. So they didn't choose anything on this, I assume. Oh no. But they had points on the last one. Okay, cool. Neutrophil, neutrophil, neutrophil. Okay, cool. Just in case there happened to be one of those really rare ones that we might get to see at some point. Good. Okay, so I see that one there, and that one there, and I think that's it.
Yeah.
Okay, so let's have a look. Yep, those two. So many. I bet you they did like 5.
Yeah. And I think again they've given a broad range of examples, because in this one I wouldn't pick anything, so
yeah,
this
I
is
agree. Yeah, no white blood cells. Okay, good, done. Okay, so then here we have 1, 2, 3, 4, and then a 5th one up there, and a 6th down here. Yeah.
Yeah.
They all look pretty similar. I think they're probably all going to be that top one that I've named.
Yeah, I think maybe the one in the top right corner is going to be
slightly different. Yeah,
yeah,
I was having the same thought just because the background is a little bit different shade compared to these. But let's click through and just see. Yep, eosinophil instead of neutrophil. Okay. All right, I think we're going to call it there.
Call it on the tutorial.
Yeah.
On to contributing to science research.
Yes, yeah. Oops. All right. Single cell 2. Okay, so that's the same as before. That's the same as before. Yeah. Thanks and happy hunting. Okay, so we have already been trained, which is great. Oh, but they're much smaller fields of view. Okay, cool. So yeah, this is what you were saying, where they've switched, instead of you're picking it out, you're just identifying. Really.
Yeah, so I think they've probably got something automated that is able to pick up the white blood cells now, based on the data that's already been provided.
Got it.
By us, the citizens in this project.
Yeah. Okay, cool. All right, so that's the one that we care about. Let's get our field guide out. It's always nice when like you're halfway there already because it's been identified. So that's great. Okay, cool. So I'm leaning towards either neutrophil or eosinophil.
Yep.
What are you thinking?
I am thinking it's the second because of the color of the cytoplasm.
Yeah. That was where I was at too. Okay. So eosinophil. I was hoping that we could just.
Perfect.
Done. Okay. Plus. Okay, cool. So this one's small and dark, so that makes it a lymphocyte. You in agreement?
I think I am in agreement. I guess I'm only just what I'm noticing is that the cytoplasm is very dark.
Yeah, exactly.
And we haven't seen the red basophil yet.
Yeah.
But
they do say that it's 2 to 3 times larger than a red blood cell.
You're right.
And this is about the same size as a red blood cell.
So I agree with I agree with you.
Lymphocyte. Oop. And we have to scroll down again carefully. There we go. Okay, cool. Let's zoom in. All right. So bigger than a red blood cell. It's got quite a lot of little dots. They are somewhat connected. I think we're between neutrophil and eosinophil again.
What are you thinking? Where's your gut going? If we're talking about, sometimes the first guess is the best guess.
Oh, you put me on the spot then. I think I would go with neutrophil. Where's yours at?
I would have gone neutrophil too.
Yeah, I think so. It's just not quite connected, but like this one over here is a little bit less connected as well and has like a really dark color with the light background. So it is really nice that they give so many examples. So I'll go ahead and do that. Okay, we'll zoom in. All right, so I think lymphocyte, because of the size again and the density of that nucleus. Is this one on mobile? Must be. Yeah.
So I was doing this on my commute yesterday, I think.
Okay, yeah. I was gonna say this would work much better on mobile than it does on this. We probably should have just done the computer on this one.
Yeah.
We'll have to think about that for the next one so that we don't have the slightly annoying pen. But
or we could do a much more complicated drawing task next time.
Yeah, that's true.
Stick to the pen.
Yeah. Well, when every nail's a hammer. Every what is it?
Every object is a hammer?
No.
To a nail, everything is a hammer?
This is not the SAT. There's a saying, like when you have a hammer, every problem requires a hammer. But there's a like pithy way of saying that I cannot remember off the top of my head.
Okay.
Oh dear. All right, I've gone with neutrophil for that. Okay, that's a lot more diffuse, but I think it's still a neutrophil.
I think so too.
Yeah. Oop. Same thing. Zoom in. I'm being influenced, I think, by that 40 to 60%, where it's like most of what we click on should be a neutrophil, probably. But that's also assuming that it's a normal spread and that they haven't just cherry picked the ones that like can't be automatically identified.
Yeah, that's true.
So that number might not be fully accurate, I guess. So again, we're between neutrophil and eosinophil. It's just because they happen to be a bit more globular that it made me think of the eosinophil here. And it's a little bit darker maybe, so I think I'm going to go eosinophil.
It's sometimes nice to mix it up, isn't it? This is like when you have a multiple choice quiz and like you answer A too many times in a row, so you start to question whether or not A is still correct. The next
one's going to be
C. even though that's not how probability works.
Yeah. Did you ever have one of those teachers in like middle school or high school that like just made every answer C, or if they were really mean, like every answer C except for one?
I did so few multiple choice questions I think that the last time I did any kind of test which had, filling in a box.
Yeah.
Like, I don't know if you had to use your pencil to circle a little oval.
Yeah.
I think that was maybe the 11 plus test that I did for secondary school.
Okay.
Like when I was 10.
Yeah. Those are called Scantrons.
Okay. It was a while ago.
You can tell we've had a different upbringing and experience with education.
Yeah.
Okay, I need your input on this one. So I've marked it as eosinophil preliminary, but what are your thoughts?
It does look like there are granules visible.
Yeah.
And so I would perhaps lean that way too.
Okay. It is nice to have a second opinion, although this is probably not how they intend for this to be done.
We're kind of creating a weird bias where it's like we're averaging ourselves first.
Yeah, that's true.
Crowdsourcing the crowdsourcing.
Yeah. Okay, I think lymphocyte.
Yeah, from size, I think we've got the size
I was gonna
down
say
now.
lymphocyte seems simple.
Yeah.
Just because of that size thing. So okay, so not lymphocyte. There's not a whole lot of background.
I think we're going to be, we're between the eosinophil and, or the monocyte, sorry. No, not the monocyte, the eosinophil and the...
Sorry, I've hidden it so you can't
neutrophil.
see it.
Do you know the difference? Do you know why
No.
one's called a cyte and one's called a fill?
I do not. I'm sure it's something to do with like Greek and or Latin.
Yeah.
You want to Google?
I'm going to Google.
Okay.
I'm going to Google while you go.
Yeah, okay. I'm on my own then. I have to make decisions. I hope no one judges me. Right, I'm going to go for eosinophil for this one. I mean, yeah, we've talked about this before, but I'm not the only one looking at these, so I shouldn't fret too much because there'll be many other people making the same decisions, and that is the power of the crowd because there will be a consensus in some way. So my one opinion is not actually that important, but I do want to do a good job, so.
Okay, so I've got a difference between a blast and a cyte.
Okay.
But I am using the Google summary of so I'm using
grain of salt.
I'm not using the AI summary from Google, but I'm using the answer picked from a website, so it just gives you that phrase.
Okay.
It says a blast is an immature blood cell. They're usually found in low numbers in the bone marrow.
Okay.
They're generally not found in the blood at all. Cytes are mature cells, such as lymphocytes. So that's the difference between a blast and a cyte.
Okay.
The difference between a cyte and a fill.
I feel like that one's going to be harder to search for because Phil is a name.
So I found a mad sci network in cell biology, an old school forum where the answer is in just text. 2007.
I was gonna say, I don't know if I trust this any more than Google's.
Yeah. Blast refers to the committed, not terminally differentiated precursor cell in a given lineage of cells. Okay, cyte refers to the terminally differentiated cell.
Okay.
So it's how far along in differentiation they are.
Exactly, yeah. Okay. Interesting.
And then fill, so that's blast and cyte.
Yes.
Fill refers to any cell that preferentially binds to a specific histological dye. So it's used to categorize differentiated white blood cells based on the contents of their cytoplasmic granules.
Okay.
I actually do you trust this source? I actually think
it
sounds very
I
proper.
do. I mean, it's clearly a scientist, but it is also like clearly their understanding, a single person's understanding, and I assume that there are no citations.
No, none at all. None at all. Yeah.
So always need to take that with a grain of salt.
Well, let's try and test it then, because we've got cytes and fills here, and do all of the fills have granules versus the cytes with no granules?
So eosinophil and basophil have granules. They're mentioned that they have granules. Lymphocyte says with no granules. Okay. Monocyte says no granules. Does neutrophil should have granules? Yep, light pinkish with small granules.
There we go.
Yeah. I love that it's based off of interaction with a dye.
Yeah.
Like how silly of a system.
I guess it's the same as, how we would categorize plants into different families and stuff before DNA.
Yeah, exactly.
DNA.
Before you had the actual tools to understand it. So, yeah. Okay, I was kind of leaning a bit, because it's so dark, towards like a basophil, but that seems wrong, just because there aren't that many. So then maybe I was like, maybe it's just like a dark eosinophil, like this one over here.
Yeah, I think that because it has those lobes, the connected shapes in the nucleus, it does look like it has that. So I would maybe go for the eosinophil.
Okay. I just want to see a basophil.
Oh wow, okay.
I don't think red blood cells are supposed to be green. Interesting.
I have noticed that the red blood cells they're looking sometimes grey. in the
Mm-hmm.
images as well, which
Yeah.
makes me think that there's been some kind of thresholding or
Processing or
processing
something.
happened to
Yeah,
the
yeah.
images,
Agreed. Yeah.
which is possibly how they then are identifying the white blood cells for
Yeah.
us to look at.
Yeah, possibly. Yeah.
Possibly. Purely speculation. Yeah.
Okay. This one's large, so I don't think it's a lymphocyte. It has quite a lot of nucleus, though. So maybe a monocyte? It's either that or the eosinophil. I keep coming back to this one on the right for the eosinophil where there's like a really large nucleus.
Yeah. I think that it is somewhere between the two, isn't it? Because the monocyte can have irregularly shaped nuclei, but like, yeah, the difference between irregularly shaped and having multiple connected shapes is
Yeah. Let's go
difficult.
monocyte. I think you've convinced me with that
Okay.
irregularly shaped argument.
Well, if
Okay.
any of the
Oh.
project owners ever watch this video, we are logged in, so you can discount all of our data if it's wrong.
Okay, this one I don't think is a white blood cell.
I think that that's debris that's been incorrectly identified.
Yeah, I think so too. Those are easy. All right, we're back to slightly harder cases. I'm thinking neutrophil, maybe eosinophil. It's always those two.
I'm feeling neutrophil.
Neutrophil.
And I can't tell if it's because we haven't clicked
Had
on
one in a while.
Yeah. It's quite small.
Yeah.
That's what's throwing me as
that's
well.
True.
It's not a lymphocyte.
Yeah.
Well,
I was gonna say again
be.
when you look at that far right one where it's like it's small but it does have those like different pockets almost inside, but they're not connected.
No.
I'm gonna go neutrophil. It's fun to me. We should have looked at the maybe we can pause here and like go have a look at some of their about because I assume that this is optical imaging. But like red blood cells are donut shaped, which is you can see that clearly here, right? But like there is actually a membrane there. It's not white. It's just that there's no nucleus, and so like the cell itself kind of just forms like a tube, an inner tube shape, and so it's like really really thin there. But obviously, whatever the imaging modality is that they're using, or like the fact that they've enhanced the contrast in various ways, means that like they're seeing white in the middle of some of these.
Yeah.
Which is just really interesting to me because it's like it should like there is a membrane there. So maybe let's pause here and go
investigate
a little bit. Just because I'm
imaging
interested
specialist
now.
wants to know what kind of imaging technique.
Apologies. It's my fault.
Well, I wonder if the staining process does something to the membrane.
Oh yeah, maybe.
What damages the membrane of the red blood cell?
Okay, so let's have a look here. So we are looking at how genes influence social behavior for rhesus macaque monkeys. They are excellent models for human social behavior, including autism spectrum disorder. Okay, so let's go down. Whole blood contains many different types of cells. This data will be compared between animals to help us determine the number and kinds of cells in the average monkey in our population, and then understand which ones are healthy and which ones may be sick. And then you can also link this to behavioral measures. Okay, fine. And then they show us a very cute monkey. And they're working in a small island off the eastern coast of Puerto Rico. See, I always think I got into the wrong job when I see stuff like that.
Yeah, why are we not going to collect, lava samples from Hawaii?
Although I don't know that I would actually want to take blood samples from like macaques.
Yeah.
But anyway, because apparently, it's 1,000 monkeys in this one island.
And how many of them have? I want to know how many monkeys we're looking at in our data set. Then is it all of them? Is it 100 of them? Is it?
Free-ranging population of more than 1,000. in the entire island, and they were brought there in 1938 from India, and ever since have been studied. Yeah, it doesn't say, but it does say that it's been understudied longer than any other primate field site in the world. So I'm guessing like a fair number of them have been sampled. It feels like this is one of those like times with observer bias, perhaps, because it's the most studied site in the world, and they're looking at monkey behavior. Like, I'm sure that they've taken that into consideration, but it does just make me wonder. Like, it'd be interesting to like talk to the researchers and hear more about their thoughts on that dynamic, essentially.
Yeah, I wonder if there has been anything done in that, because I know that there are studies that show that cows, for example, will change their behavior if there is a human around.
Did you know that mice do the exact same thing? One of our main model systems in like all of scientific research, they change their behavior if a human is in the room. And not only that, they change their behavior if that human is male versus female.
I didn't know that, but
yeah.
of course it's the case, and of course no one's really thought to check in advance. Yeah.
Well, yeah, I know it came out when I was in grad school, so like, a good number of years ago now, but like at the time it was like, oh no.
That's all prey animals kind of do that though, right?
Yeah.
Yeah.
I think a lot of them do, yeah. So there's been a push recently, I think, in like the mouse facilities to try to come up with strategies to like alleviate some of those variations, essentially.
It's just only one person can look after them then. Or
well, I think
maybe there's a favorite that you can identify by behavior.
Yeah, exactly. No, I think the like real fix is going to be to remove the humans.
Yeah.
Eventually, to have like automated cages and things like that. And I think some places are moving more towards that, but it will take time. So
yeah.
But yeah.
Well, the humans are biased intrinsically, right?
Yeah.
Like we are the.
Yeah, we're the problem.
Yeah.
We're the problem and the solution sometimes. But okay, so they do daily behavioral data on focal animals, on specific animals. So they do talk a little bit about it. The animals are used to humans being on the island, so human presence does not seem to impact their behavior.
Citation.
A citation needed, yeah. Okay, so they're looking at the amount of time it spends alone with other animals, grooming, eating, etc., and they record that, and then they compare it to other metrics of health, such as blood counts. Okay, good. Right, then blood typing. Whole blood is collected from a subset of animals living there each year. Whole blood is smeared across microscope slides and is stained to better distinguish the types of cells in the blood, which makes sense, like this was pretty clearly stained. They use a microscope to view or take images of individual microscope slides that have monkey blood on them. And they count the number and types of red blood cells, both red and white, and platelets. So they're doing a bit more than what they've asked us to help with here. And then they classify them. Okay, and they assess for unusual red blood cell shapes.
Your favorite?
Yes, I do like red blood cell shapes. Although I doubt that any of these monkeys have sickle cell disease. Unusually low or high numbers of white blood cells, which signals an infection, and unusually high numbers of rare white blood cells, which signals disease. Okay, so they don't really talk about how many samples, how many monkeys have been assessed using this. And they don't really talk about the microscopy other than that it is just
Your
died.
favorite part of the project.
I know, like one sentence. They just, two, I guess. So microscope slides, and then they use a microscope to image it.
I saw that there's a paper that they've published, so the methods of that might tell you exactly what you want. But...
And there's a presentation too. Let's not bring the whole. Oh no, it's a video. It's the thing that you didn't want. Undo. Okay, I think we have to stop there.
It's the end of the day for us here. It's the end of the week as well, so perhaps we've reached our limit on how much we can contribute to scientific research.
Yeah. We'll have to have a look later at the different microscopy types used for that project.
We will. Yeah, you're going to have to deep dive on that
Yeah,
paper.
don't worry.
That'll be our homework for the week or for the
Yeah.
few weeks that we're gone.
Yep.
Yeah.
So next stream is in 2 weeks?
Yeah, the 11th of July, same time, 4 p.m. British Summer Time.
Yep.
Yep.
If you have any suggestions of projects that you would like to see us attempt, please let us know. Ideally in advance so that we can make sure we have the correct tool next time. And if you are aware of any researchers that you think might want to get involved or be interviewed as part of this, let us know, because we're thinking to expand a little bit. All right.
That's it.
Thanks for watching.
Yeah. See you next time.
Bye.